Welcome to the Deep Dive, your shortcut to being well-informed. Today, we're looking at something, well, quite paradoxical, tuberculosis.
Yeah.
Even with all our science, TB is still the single deadliest infectious killer globally.
It's staggering, really.
Yeah, just this year, we're talking 10.8 million cases and 1.25 million deaths. Now, compare that to something like smallpox.
Which we wiped out with vaccines.
Exactly. Completely eradicated. So the big question we're diving into today is, why is TB still such a massive problem? Especially when we know T cells are absolutely key for immunity. We're going to unpack this whole complex world of T cells and Mycobacterium tuberculosis, or MTB, and really look at the knowledge gaps that are stopping us from getting that effective vaccine.
And that really is the core issue, isn't it? We know T cells are vital for fighting TB. We've known that for a long time.
Yeah.
But how they actually protect us, the precise mechanisms, well, that's still surprisingly unclear.
And MTB itself makes it hard.
Definitely. It's got this huge genome, right? Around 4,000 potential antigens, targets for the immune system. Plus, it's co-evolved with humans for, millennia.
So it knows our tricks.
It's developed some very clever ways to evade and even sort of manipulate our immune responses. It's a very sophisticated pathogen.
But like you said, there is some light here. There have been some pretty landmark discoveries recently, especially in non-human primates, NHPs.
Yes, those studies have been incredibly encouraging.
They've shown vaccine strategies, things like using BCG, the current vaccine, but giving it intravenously or intrabronchially, or even a completely new type, this rhesus cytomegalovirus vaccine.
The rhCMV vector one.
Right. And these have actually achieved sterilizing immunity in some animals, which means what? Complete protection?
Complete protection, yeah. The animals just don't get infected. It's the gold standard, really.
And that's translating hopefully to humans. I heard there are clinical trials.
There are. It's really promising. There are currently 8 phase II or phase 3 clinical trials for new TB vaccines underway. So progress is being made.
Okay, so let's rewind a bit. Start at the beginning. MTB gets into the body, usually the lungs. How does the immune system first react? What's the initial encounter like?
Right. So MTB is an intracellular pathogen. Basically, it likes to live inside our cells, specifically phagocytes, the cells designed to, eat things.
Like Pac-Man cells.
Kind of, yeah. So when you breathe in the bacteria, it gets snapped up pretty quickly by alveolar macrophages. These are the immune cells just sitting there in the air sacs of your lungs.
The first line of defense.
Exactly.
Yeah.
But MTB is tricky. It replicates inside those macrophages, and then it can spread to other cells too, like monocytes that get recruited from the blood to the infection site.
The reinforcements.
Right. And those monocytes then change. They differentiate into what we call monocyte-derived macrophages. Now, here's a really key point. These two types of macrophages, the resident alveolar ones and the new monocyte-derived ones, they handle MTB differently. Ah, okay. How so?
Well, it seems T cells are pretty good at helping control MTB replication, even promoting killing inside those alveolar macrophages. But they seem to have less of an impact on the MTB inside the monocyte-derived macrophages.
That's a really important distinction. So given that, what are the T cells actually doing in this early phase? And how do they even get, activated or primed to recognize MTB? Is it just a direct meeting?
Yeah, it's not quite that simple. Both main types of T cells, CD4 plus and CD8 plus 8A, are absolutely essential.
Okay.
The current thinking on how they get primed, that initial training is quite interesting. It seems MTB-infected monocytes, or these related cells called monocyte-derived dendritic cells, or DCs.
Right.
They travel from the infected lung tissue to the nearby lymph nodes, the ones that drain the lungs.
Okay, makes sense. Immune HQ.
Sort of, yeah.
Yeah.
But interestingly, these cells that migrate there, they aren't actually that great at priming the T cells themselves.
Oh, so how does it happen then? It looks like MTB antigens. So bits of the bacteria, not the live bug itself, get transferred to other DCs that are already living in the lymph node. These resident DCs are much, much better at presenting those antigens and activating the naive T cells, both CD4 plus and CD8 plus dot.
Like a handoff.
Exactly, a handoff to the specialists. And then once those T cells are primed and activated, they travel back to the lungs, ready to hopefully fight the infection.
So we've got the initial response, T cells getting primed. Now, digging a bit deeper, what kinds of T cells does MTB mostly trigger? Is it just one type or a mix? And critically, how do things like a person's general health affect whether they actually get sick with TB?
MTB mainly induces CD4 plus T cells that produce a molecule called interferon gamma, or IFNO. We call these Th1 cells.
The classic killer helpers, right?
That's the classic view. Yes, they're really crucial for dealing with intracellular things like MTB. But there's more and more evidence that other types are also important, like Th17 cells, which produce interleukin-17. They're more involved in recruiting other cells, and even cytolytic T cells, ones that directly kill infected cells. It seems to be a team effort.
And what about those risk factors?
Right. We definitely know certain things dramatically increase your risk of progressing from just being infected to having active TB disease. HIV co-infection is a big one.
Yeah.
And also treatments that block TNF, which is a key immune signaling molecule. For those, we have at least some idea of the mechanisms involved, how they impair the immune response to MTB.
Okay.
But then there are other huge risk factors like malnutrition or diabetes mellitus. And for those, honestly, the underlying mechanisms, why they make TB worse, are still pretty unclear. That's a major gap in our knowledge.
Which brings us back to those NHP studies. If they showed such amazing results, sterilizing immunity, why aren't those vaccines like the intravenous BCG or the rhCMV1 already being rolled out for people?
Yeah, there are some significant practical hurdles. For intravenous BCG, there were safety concerns that came up in early phase I studies, actually in cancer patients, not TB.
Oh, really?
Yeah. Giving BCG straight into the bloodstream, well, it raised flags about potential widespread dissemination. The vaccine bacteria spreading too much. It's different from the usual skin injection.
Okay, that makes sense. And the CMV vaccine?
With the rhesus CMV vaccine, the big question mark is pre-existing immunity. Most people, a very high percentage globally, have already been infected with human CMV.
Right. It's really common.
Exactly. And we don't fully know how that existing immunity would interact with a vaccine using CMV as the delivery vehicle. Would it boost the response, interfere with it? Could it cause safety issues? Those are still open questions before it can move forward in humans.
Okay, so let's shift focus a bit to where the battle actually happens in the lungs, these structures called granulomas. They're fascinating, but also seem problematic. What's going on inside them, and why does it seem like T cells are kind of kept out of the main action?
Granulomas are these organized balls of immune cells that form around the MTB infection. It's the body trying to wall off the bacteria. But inside, we see this thing called spatial segregation. Meaning the MTB-infected macrophages, the cells actually harboring the bacteria, tend to be stuck right in the center, the core of the granuloma. While the T cells, which are supposed to kill those infected cells, are often found more on the periphery, sort of circling around.
They can't get in?
It certainly looks that way. We see this pattern in mice, in macaques, and importantly in human TB granulomas too. And the thinking is this physical separation really messes things up for the T cells. Because for T cells to effectively kill infected cells or help macrophages kill the MTB inside them, they generally need direct, close contact. If they're stuck on the outside looking in, their effectiveness is likely impaired.
So is MTB deliberately keeping them out? Is it an active strategy by the bacteria, or are other things causing this separation?
It looks like MTB is definitely playing an active role. It has a whole arsenal of tricks to mess with T cell responses.
Like what?
Well, it can interfere with antigen presentation. That's how the infected cell signals, hey, I'm infected, to T cells. MTB can block the cell's machinery for processing its own bits. It can divert MTB proteins away from the presentation pathway. It can even make the cell display fewer of the MHC molecules that T cells recognize.
Wow, okay, so it hides the signals.
Exactly. It also tries to stop the host cell from dying, which would normally release antigens. And it can directly mess with the T cells themselves, interfering with their activation signals, the TCR signaling, and triggering inhibitory pathways. Things like IDO, TGF4, IL-10. These basically tell the T cells to calm down or stop working.
It really sounds like MTB has evolved this incredibly sophisticated defense system. What about our own immune system's controls? Can they sometimes backfire in TB?
That's a really critical point. Our immune system has its own checks and balances, these co-inhibitory receptors, sometimes called checkpoint inhibitors like PD-1 and TIM3. They're normally there to prevent excessive immune responses and damage.
Like the ones targeted in some cancer therapies.
Precisely. Blocking PD-1 can be great for unleashing T cells against cancer. But in TB models, doing the same thing can actually make the disease worse.
Really? Why?
It seems to cause too much T cell activation, too much inflammation, which damages the lungs. It highlights this key issue in TB. Often, the damage isn't just from the bacteria, but from immunopathology, the immune response itself being too strong or damaging.
So trying to boost the T cells can be a double-edged sword.
It absolutely can be, especially without good antibiotics on board. Interestingly, blocking a different checkpoint, TIM3, has actually shown some benefits in TB models. So it's complex. You have to target the right pathways. And all this leads to some really big unanswered questions about T cells in granulomas. Like first, why are they stuck on the periphery? Is it problems with the blood vessels they use to get in? Maybe not enough of those specialized HEVs, although we know HEVs generally correlate with better immunity.
Or maybe the chemical signals are messed up.
Could be. Or physical barriers, like the dense extracellular matrix, the sort of gristle of the granuloma blocking their path. Second, even if T cells get near, do they actually lock onto the infected cells? Or are they getting distracted by uninfected bystander cells nearby that might also be presenting some MTB antigens?
That's an interesting wrinkle.
Third, how exactly does MTB manage to hide from T cells even when they are nearby? Studies show only a tiny fraction of the MTB-specific T cells in the granuloma are actually activated. And even if you boost activation, the bacterial numbers often don't drop much, probably because of that spatial separation.
Right.
And finally, there's the role of tissue-resident memory T cells, TRM cells. These are T cells that live long-term right there in the tissues, like the lung. They're thought to be really important for immunity, especially from vaccines delivered to the mucosa. But where exactly are they within the granuloma? Can they access the infected cells? We just don't know.
Okay, we've definitely laid out the challenges and the big unknowns. Let's shift gears to the really exciting part now. What are the new tools, the cutting-edge technologies that scientists are using to try and answer these questions and crack the TB immunity code?
Yeah, this is where things get really fascinating. The technology now is just incredible. First off, they're spatially resolved omics analyses.
Okay, omics meaning like genomics, proteomics.
Exactly. Genomics, transcriptomics, proteomics, looking at all the genes, RNA, or proteins. But the key here is spatially resolved. These techniques let us see which molecules are active in which cells and where those cells are located within the actual tissue structure of the granuloma.
So it's like getting a molecular map laid on top of the physical structure.
Precisely. Technologies like spatial transcriptomics or multiplexed ion beam imaging give us unbelievable detail about cell states, who's talking to whom at a molecular level, all while preserving that crucial spatial context. We can see, for instance, that the necrotic dead center of the granuloma is often immunosuppressive, while the edges might have more activated immune cells. Different neighborhoods within the granuloma have different rules.
That's amazing. It's like seeing the battlefield in high def, understanding the terrain. What about watching the troops, the T cells, actually move around?
For that, we have multiphoton intravital microscopy. This is just mind-blowing stuff. It lets us literally watch, in real time, dynamic processes inside living tissue.
Inside a living animal.
Yes, often in animal models. We can visualize T cells migrating, crawling around, interacting with infected cells, or getting blocked by barriers like that extracellular matrix we talked about.
Wow.
It's essential for understanding why T cells get stuck, why the spatial segregation happens. It's technically very challenging, of course, especially doing it safely in live lung tissue with MTB, which requires high biosafety levels, BSL-3, but the insights into actual cell behavior are game-changing.
That brings up another huge challenge. MTB has, 4,000 potential antigens? How on earth do you figure out which specific ones are the right targets for a protective vaccine? Finding that needle in a haystack.
That has been a massive hurdle for decades. Traditional methods often relied on just one readout, like IF-no production, which might miss other important protective responses, and antigen presentation varies. But now, we have much smarter approaches. Well, researchers are studying large groups of people, human cohorts, who have different outcomes after MTB exposure. Some control it, some get sick. By comparing their immune responses, we can find antigens that are strongly associated with protection.
Learning from people who naturally fight it off well.
Exactly. We can also now directly identify the specific bits of MTB, the epitopes, that are naturally processed and presented on the surface of infected cells using really advanced mass spectrometry.
So, seeing what the immune system actually sees.
Precisely. And we can analyze the T cell receptors, the TCRs, on protective T cells to figure out what they're recognizing. Plus, people are using AI to predict structures, and new tools like DNA barcoded libraries of epitopes combined with single-cell sequencing to screen possibilities much faster.
It sounds like we're finally getting the tools to really zero in on the right targets. What about the animal models themselves? Are they getting better at mimicking human TB?
That's another crucial piece. We need models that better reflect the human disease if we want preclinical tests to be more predictive. And yes, there's progress there too.
How so?
We now have mouse strains, like the C3-HBFEJ strain, that actually develop lung granulomas that look much more like human ones, with that necrotic center. That's a big step up from standard lab mice.
Okay.
And what's really exciting is the use of genetically diverse mice, like collaborative cross or diversity outbred mice. They capture much more of the genetic variation you see in humans, which is critical for understanding why some individuals are more resistant than others, and how vaccines might work across different genetic backgrounds.
Moving beyond just one specific mouse type.
Right. And maybe the most fascinating development here is humanized mouse models. Things like the velocit mice. These are mice engineered to have key components of the human immune system, particularly so their T cells recognize antigens in a way that's very similar to human T cells.
So you could potentially test a vaccine candidate in these mice and get a much better idea if it will work in people.
That's the hope. It could bridge that gap between standard animal models and human trials much more effectively. And there are other cool things emerging too, like mass spectrometry imaging, which can map all sorts of different molecules, lipids, metabolites, drugs directly in tissue slices at really high resolution without needing specific antibodies, and the development of 3D organoids.
Miniature organs in a dish.
Basically, yes. Tiny self-organizing structures made from human cells that mimic aspects of lung tissue, sometimes even including blood vessels. They let us study cell interactions and tissue architecture in ways you just can't in a flat petri dish. And underpinning all this, really important, is the increasing push for data sharing.
Making all this information available.
Yes, through public databases like the Human Immunome Project, IMPORT, the Immune Epitope Database. Sharing these huge, complex data sets is absolutely vital to speeding up discovery by the whole research community.
Okay, we've covered a huge amount, the complexity of TB immunity, the roadblocks, and now these incredible new technologies. Looking at all this, what really stands out to you about how these tools are changing the game in the fight against TB?
I think what's truly transformative is the level of detail we can now achieve. We're moving beyond just observing correlations. This type of T cell is present when things go well. Now we can really drill down into the mechanisms.
The why.
Exactly. Why are T cells spatially segregated? Which specific molecular interactions are preventing them from working? Which antigens trigger truly protective versus non-protective or even harmful responses? Answering the why lets us design interventions, especially vaccines, much more rationally. This shift from observation to deep mechanistic insight, it's just revolutionary for the field.
It really sounds like it. Despite MTB being such a clever adversary and our own immune system being so complex, these technologies are giving us an unprecedented look under the hood. It feels like they're not just filling gaps, but actually changing how we think about designing a TB vaccine.
Absolutely. If we connect this to the bigger picture, it's not just about inducing any T cell response anymore. It's about inducing the right T cells, maybe specific types like TRM cells, ensuring they have the right functions like cytotoxicity or cytokine production, and critically making sure they can get to the right place inside the granuloma next to the infected cells.
That level of precision is finally within reach.
That's the goal, to tailor the vaccine-induced immunity to overcome MTB's specific evasion strategies and the challenges of the granuloma environment. That's how we'll hopefully achieve truly effective protection against this incredibly persistent pathogen.
So here's a final thought for you, our listener, to consider. If we can now map the intricate dance between immune cells and microbes within a TB granuloma with its kind of stunning detail, what other long-standing puzzles about our immune system might these technologies unravel? How could this microscopic view transform our fundamental understanding of health, disease, and immunity way beyond just TB?